Actually, you should start recording there. Yeah, welcome. I was just over on your stream. Welcome to the rating party. I didn't know you were going to be like there, like just like that. I literally was having just audio problems, fixing it up. We're talking about school right now. But welcome, Raiders. Hello, casual Phoenix? Um, let's see, what are we going to do today? See, this is the question I had for myself earlier today. Let's see. Whole genome sequencing. So yeah, I did kind of say on Discord that I wasn't sure if I was going to stream today, but things worked out. I am here, obviously. We're going to do a stream. I'm unprepared, so we'll just talk about some things. So today I wanted to talk more about gene editing, the real and fictional, genetic engineering and sci-fi and medicine. So last time, who was it? I believe it was... Let me double check. It was on the Discord, I think. I think it was David, who I don't think is here right now. David T. Gregory suggested... Where is it? He had a specific thing, and it was all about cloning. And I thought it was Metal Gear Solid. That's it. So I definitely want to look at Metal Gear Solid. And related, Stargate. Manogard, I know you're here. Because it kind of sounded like there were very similar things there. And I kind of count these in sort of gene editing. Because both in Stargate, the Asgard, and it sounds like in Metal Gear Solid, they were doing a lot of tinkering with genes when they were doing their cloning. So I kind of wanted to look at those, and we could talk about some of that. So that'll be fun. But first and foremost, a couple new things. There should be some new chat rewards. Yes, there are new chat rewards. Not a lot. So you can check those out. Still working on that. Yeah, everything looks about right. There's limits on things, too. There also should be a little... Oh, I'm not seeing it. There should be a new little icon for the chat rewards, too. But maybe it doesn't show up on my stream software. Oh, that's okay. All right. Oh, yep. That was one of them. I hope the sound worked for that. I had to say be mean because I had to do be mean because Twitch kept getting angry at what I had it as. Apparently you can't have like mean sounding language as channel rewards. So I had to be very, very vague about what was actually going on. So sorry if that's vague. You can just try it out and see what it does. Although if you checked it out on Discord, you already know. All right. Let me go ahead and scooch some of this down. So before that, though, related to stream, something that I feel like is a sort of a stretch goal, very long stretch goal, and I'm not confident enough to actually put it up on stream or on...
Loser! You're a loser! Are you feeling sorry for yourself? Well, you should be.
Yeah, so that scared me. Yeah, so I couldn't call that. I wanted to call it "You're a Loser." Twitch didn't like that, so it became "Be Mean." Thank you for redeeming your chat rewards, I guess. Yeah, so if you ever happen to find... Hold on. Green screen Hank Hill. If you ever happen to find anything where there's a green screen like this, I can make it a chat reward. I taught myself how. It was very easy and it was very fun. So I'm excited just to add more. If nothing else, just to scare myself. All right, Team C, go away. Okay, stretch goal for stream. I don't know how to set this up. So I've already had a number of subscribers, which has been awesome. Super, super thankful for that. It's not why I stream, but it's always nice to feel wanted. But if I were able to get about 50% of the cost of one of these, what I would want to do is do one of these whole genome sequencing tests. I could do that on stream, but it's going to be spitting in a tube, so maybe that's not super interesting. But the cool thing is you can get your data out of it. So what is this? I think I have to actually pretend buy one. So this is more than what I was showing on stream last time. So last time on stream, it was a SNP test, SNP test. It does say 70% off. It's always been 70% off. I've been watching this company for many, many, many months now because I really want my genome sequenced. And it's always $2.99 for their normal one. It's just always that. Summer sale, sure. It was also the spring sale and the winter sale and the fall sale. So last time, did I download it? Do I have it somewhere? Yeah. So last time we looked at my DNA. Remember, we have these Control plus. Here, you can actually see it now. Right, so for each of these little identifiers on the chromosome at this position, I have two alleles, two sequences, one from my mom, one from my dad. These are single bases. These represent something like 0.001% of your DNA. Granted, these are often the bits of your DNA that matter. Most of it's relatively the same, person to person, above 99% similarity between any two people. That said, it's not all the information. And so the idea between whole genome sequencing is literally getting all of that information. Too bad we live in a pandemic microplastic hell. Sorry. I just you're right. Just throwing in microplastic between pandemic hell world, very appropriate. Anyways, too bad we live in a pandemic microplastic hell world, so your students will probably never get to extract their own DNA lab ever again. Yeah, that kind of sucks. Can you not look at probably not even their cheek cells under the microscope anymore either, right? Because that's still spit related. Although teenagers are always swapping body fluids, so it doesn't really matter. I don't know. Yeah, you're right. More of those little tubes will be microplastics. I use strawberries. Strawberries are good stand-in for teenagers, I have found. No, I know what you mean. I kind of forget your background in science because I know you work in or have worked in the agricultural side. I don't remember if you've ever been in a wet lab, a research lab, not a research lab, a microbiology or a molecular biology lab. We go through so much plastic. It's kind of ridiculous. It's frightening how much plastic we just every day, just plastic everywhere. That said, well, yeah. Hello, Megan. Yeah, the number of tubes and pipettes you go through in a day it's terrifying. It really, really is. How many hundreds are you going to do this week? Who knows? You've put your own cheek cells on prepared slides. Nice. Yeah, that's always fun. Let me see if what I have to show. So, okay, whole genome sequencing. What's the difference? Hold on, open up a program that I hope will not freak my computer out while streaming. It's opening in the background. You worked in between. You had to waste a lot of plastic bags and tubes and samples and feed stuff to the. Okay, yeah. I see. I kind of got that sense. Yank. All right. So we're looking at a genome browser. But open session? Open session. What do I want to look at? This is the wrong genome. This is the wrong genome. We're looking at the human genome. We got to look at the fly genome. All right, whole genome sequencing, what you get back is, give me a second. Ugh. Fine, index the file. Please just let me do this. Run batch script? Nope. Four flies only? What? Index. Input file. This file. Run. False. Okay, well, I thought I was going to be able to show this. Guess we're going to call it no. Whatever. All right, I thought I wanted to show the individual reads, but essentially, like whole genome sequencing, whereas, see, I've been zooming in this whole time, whereas SNP testing is like checking these individual sequences down here for differences. Right, so like this A is maybe a T part of the time. Instead, we're looking up here at the top. Track height. This is 200. And as I zoom out, I can see that those little sequences go away, and it gives way to these massive sets of sequences. Also auto scale you. So right now we're looking at about 5 million, or about 6 million bases, 7 million bases, as I sort of like scoot along the genome like this. This is more or less what I want. Full coverage of my DNA, so that every single gene, so if I zoom into this 18W gene, whatever this is, we can see the full sequence of it. Even though, oops, I scooted away. And I lost it. I went somewhere else. Because, you know, genes aren't represented by single bases. Like this gene right here happens to be about 63,000 bases long. A normal SNP test probably grabs maybe 1, maybe 2, maybe if you're really lucky, 3 in sort of a gene span of this size. What would be really cool is full coverage across the entire thing. I do want to try one more time to load something from a file. I'm just kind of surprised I don't have anything. I normally have a bunch of just random junk. BG. Nope. What's this one? Nope, guess not. Okay. Anyways, the cost of this, let me buy it. Let me put it in my cart. So they say it's, you know, whoa, nope. Go away, honey. So they always say it's $1,000. It's not $1,000. What it really is about $299. They want you to buy this like monthly sort of cost. So that was the lifetime purchase. But all you really get with it are updates to the reports, different like services that they do, genome exploration tools for their subscription. But that's something that I literally do for work, so I can figure that out. So I'd probably get the quarterly, cancel it immediately. So I need about $300. I would say that through the course of streaming, if I make 150 like dollars via bits, donations, subs, however many months that will take, I'll just go ahead and buy this. I won't spit in a tube on stream. I'll send it off, but we'll open up the raw data. Yeah, we can get the raw data, and we can like work through it. See if I have anything weird in my DNA. Oh, you were trying to say for flies, only like an exclusive club. Ah, then your kids terrorize you. I love flies. Flies are great. Anyways, long-term stream goals. This one right here. Okay, what do I have back here? All right, so what did I want to look at? So I got to look up this Metal Gear Solid cloning. Because I love seeing what people say is cloning. Right, did I get that right? Metal Gear Solid, is that what I said before? Metal Gear Solid. Yeah. I guess I didn't know that they were doing cloning in Metal Gear Solid. And this is the Metal Gear Wiki, apparently. So I would hope that there's maybe some better references exactly, because this is so small. I think even probably this sentence right here, the term clone is derived from the ancient Greek word clone, or twig, referring to the process whereby a new plant can be created from a twig. That's interesting. I didn't know that. But this sounds like it's taken directly from Wikipedia. Oh, they just have, there are clones. Yeah, see, I wanted to see how they were doing cloning. It's kind of interesting just because... Oh, hold on. Oh, my computer's being slow. I guess it's a good time to open my water. There we go. Oh, hey, there's a YouTube video. Let's watch this and get some information. Maybe we'll learn something. That was very loud for me. Okay. If this is true, I can't verify because I've never played a single Metal Gear Solid game. Is this spoilers for the game, or is this well known? Who's Liquid Snake? Liquid Snake. Liquid Snake. Huh. What about Gas Snake? I don't know. I'll search it. Gas Snake. Why isn't Solidus called a Gas Snake or Plasma Snake? That would be the logical progression, right? Well, it looks like other people had the same question. Also, welcome. No worries. I was very up in the air whether or not I was streaming tonight, so it was sort of a surprise. Alt Miss Frizzle and Manogard already had chided me on being kind of, I don't know, quick on the draw, just all of a sudden streaming. Incredible soldier genetics. So, okay, well, hold on. Yeah, so I don't know. Okay, I'm gonna just go search that. Metal Gear Solid. What did he just say? Soldier genetics? Oh, soldier genes. The autocorrect. Oh, what are the soldier genes? Hey, you're creatures of habit. Yeah. I know. Thank you for coming. Thank you for being so quick. You both logged on as soon as I went live. I usually go live a little early so people have a chance to, come by. I was here turning off lights, turning on lights, getting water, and you were already here. Let's see. What are soldier genes? What do they do? How are they different from regular genes? I mean, anyone can shoot a gun and kick ass with proper training. What difference does it make? I mean, that's a good question. Oh, this person has an answer. The soldier genes in MGS fiction are specific genes in Big Boss that apparently give him the edge over other humans. Not by default, but his maximum level of development and in combat related skills and fitness is, in theory, better than everyone else's, probably by chance. Okay. That's actually not, I mean, that's not terribly wrong. So presumably, most things about our capacity. See, I don't want to say capacity for a certain skill because humans, on average, very similar to each other. But some people are better at things than other people, on the whole. So maybe. Okay, so if this person was for some reason particularly like they had very steady hands, were very good at judging distance, very good at aiming, had like a very very quick trigger finger, like very quick I don't know, like muscle movements or something like that. That could be genetic, sure, why not? I don't think we've seen. Okay, soldier S&Ps. Probably. Oh, look at this. Genome-wide analyses of psychological resilience in U.S. Army soldiers. Okay, so that's not ability, or at least combat ability, but certainly psychological resilience is probably very important for a soldier. So I won't. Oh, this is actually really good. This is very related to what we were just talking about. So when we look at genes, like my genes right here, the way we know why some of these, so for example, RS6696281, why we know that having two C's is really good for some reason or really bad, is through a type of study called a GWAS, which is a genome-wide association study. Basically, what the researchers do is they collect a bunch of people together, have them take a bunch of surveys or do a bunch of tests, like physical or mental or something like that, or they follow their health, basically any question you have, and then do a SNP test, a single nucleotide polymorphism test like this one, and look around. And let's say you had a population and you were really interested in, for example, psychological resilience. So what's your population? It's U.S. Army soldiers. Sorry, I'm getting an Amber Alert. So your population is the U.S. Army soldiers. What are you measuring? You're measuring psychological resilience. I don't know how they measure that, but clearly they have some measure. And people fall on like a gradient, right? Some people have little measured psychological resilience. Other people have lots of psychological resilience. You could also do something like measure lactose tolerance, right? Some people have no lactose tolerance. Some people have great lactose tolerance, right? So you can measure all sorts of things. So now that you have your population, your measurement, next you go to the genome and you look around and try and see, okay, what in their genes is associated with this trait, either positive or negative, right? So maybe there's a particular RS number, right, that is associated with psychological resilience, like I just said. So maybe this right here, maybe most of the participants in the study who had C's at both loci show up. Sorry, I'm reading the thing now. Maybe the people who have both C's are more likely to have psychological resilience. The paper is behind a paywall, but you sent me the PDF in Discord in case you want to end up reading. Is this in... oh. Struggling? Oh, thank you. Thank you. Yeah, let me link to this in Discord if you want to look at it too. I'll put this in the primary literature. Oh, never mind. Thank you, Brian, for being a good mod. Brian already did it. Okay. So, thank you. Yes, that's what the primary literature section is for. So they did some measures, blah, blah, blah. Okay, so now here's something that's really, really cool. So this is called a Manhattan plot because it looks like the Manhattan skyline, right, with all the skyscrapers and stuff. And they're basically asking, at each point along the chromosome, there are many, many SNPs. So each one of these dots corresponds to each one of these lines, right? And it basically just asks, okay, what is the likelihood that that SNP is associated with a certain trait? Okay, so on the y-axis here, it's minus log 10 p-value. All that means is the higher up it is, the more significant, not more significant, the more confident you can be that the results are significant. So they have some arbitrary, semi-arbitrary line here. And you can see that there's like one spot right here. This right here that goes above this line, meaning that they are very, very sure that these dots above here are likely associated with psychological resilience. And I'm just looking at this table right here. It looks like there's also population metrics, which I know are important. I just don't know how they measure them. So basically everywhere else in the genome not associated, or at least not significantly associated with psychological resilience, but it looks like there's one spot that was. This is on chromosome 4. Yep. So we zoom in on chromosome 4, and there are now it's just like a zoom in. Same dots as before, just sort of broadened out. And down here at the bottom, we also see a gene. So what direction is that? Okay, that gene is going, wait, on camera, it's going, whoa, this way. My camera's reversed. It's going that way. So it looks like this association is in this regulatory region of this gene, DCLK2. DCLK2 gene. Let's look up what that is. The double cortin-like kinase 2. What does it do? Bind microtubules and regulate microtubule polymerization. Mouse studies show that the family member of this gene family shares function in the establishment of hippocampal organization. Interesting. So there's like a brain type function for members of the same family, so that's good. They give an RS number. I wonder if that's on my DNA. Okay, it might not be. RS 4260523. Oh, okay. I have it. Well, I mean, of course I have that base. The question is whether or not I have like a resilience phenotype. I'm homozygous for it. Oh, in fact, look at this. I'm a very homozygous at this region. So which means that I have the same sort of DNA or the same allele from both my mom and my dad. But it's this middle one. So I just wonder what that is. Okay. Did they say what it actually, it's a marker, but what is it actually? They might just be they might not have said what it is specifically. They might have just been looking at whether or not it was the same in individuals. I would really hope that they mention exactly what base it is. Anyway, so this is all to say that, okay, sure, why not? You could call this a soldier gene. When was this published? This was published in 2019. Okay, so not so far away. A significant locus. Yep, 4 single nucleotide polymorphisms. But wait, is this a different number? This found for 1 locus. Oh, there's another one that was just sort of high, but not super, super high. So I'm not actually used to reading these types of studies, so I actually don't know if it's common to put the base, or if they're doing it, but not just in a way that I don't know. Please tell me what it is. I want to know if I have the soldier gene. I want to know whether I'm worth cloning to be a soldier. GWAS. Okay, that's resilience. SNPs. Let's see. Hippocampal volume. Hello, Champ Cal. Jango Fett had the soldier gene? Yeah, why not? I don't understand Star Wars. Are they canonically like humans? Please tell me supporting information. Brian, was there supporting information that was easily accessible for this? Yeah, they did clone an entire army, and I don't know if did they ever discuss like what they did to them besides just make them, grow really fast. You'll try to find it. Thank you. I just want to know what the base is. I mean, I guess we can look up this particular SNP. So let's look this up on SNPpedia, SNPedia. Maybe they'll say something worthwhile. Say something interesting. Oh, they mentioned the study. Okay. Oh, here we go. Oh, interesting. So down here, oh, it's very hard to see for everyone. Nope. Control plus. So this plot right here shows the alleles, so AA, AG, and GG. Oh, you sent it on Discord. Thank you. I'll get to this first. Oh, the mutations are in the tables. Perfect. Well, we'll reveal that afterwards because already looking at this, I can tell that I have a not unique but rare genotype. So it looks like in Utah residents with Northern and Western European ancestry. I'm not in Utah, but I do have Northern and Western European ancestry. So actually that's about... Yeah, so the Northern European is about what I am. So that's the closest. So it looks like a very... Oh yeah, only 13% have a GG, which is what I have. Most people of my vague ethnicity are either AG or AA. It looks like most people worldwide, at least of the ones that were studied here, have AA or AG. So that's interesting. Whatever for whatever it's worth, I have a more rare genotype. But you linked the supplemental tables. Thank you. I have to open it Word. Okay. So. Okay. So these are the SNPs and the p-values. So these are the significant ones. In blue / bold are the suggestive ones for the resilience. They're all located near this DLC or DCLK2, this gene that might have a function in brain organization, brain development, something like that. So let's cross-reference this with what I have. So they're saying right here, allele 1 A, allele 2 G. So actually I don't have the soldier genotype, at least with that. There's no guarantee I have these others, but I will look them up regardless. Nope. Uh-oh. Not that. Nope, not that one. Do I have any of the soldier genes? No. Well, not that I don't have them, it's just that I don't have data for them. But again, if I got my whole genome sequence, we could figure this out, because we could look at every single locus. There was another one that they kind of ignored. Why did they ignore that one? RS100. Oh, because of the significance value. It's slightly closer to 1 than the other ones, although it's still significant. There's a lesson to be learned in understanding the difference between something that is significant and something saying that you are very confident in your results. All right, well, whatever. At least I have the one. Okay, cool. I won't close that. Okay, thank you, Brian, for that. It's very useful. Is this the same one? Yeah, it is. Oh no, it's not. Some other ones. Okay, they're just going deeper and deeper with like the covariates, which are, like other things that might contribute. I don't want to look into that. That's going to be too confusing, and I don't understand how they do that. All right, but thank you very much. Okay, so, well, that's cool. That was very related, and I got to show off a Manhattan plot, a plot I've never made, but I think is really cool looking. Okay, back to this video though. Wait, so far so good. Well, I would say we can go like iffy on the like soldier genes. Like, of course, stuff like that could be genetic, but it's probably not quite as determinate as the leaders of Project L'Enfant Terrible would say. I think you actually want to be pretty careful about which cell you choose. See, this is something that Dr. Z would know. If people remember from when Dr. Z was on, they did somatic cell nuclear transfer to clone pigs that they were then CRISPRing or using CRISPR-mediated gene editing to make more human-like, the organs at least. And I meant to ask, I might send him a message on Discord and just ask. So whether or not like any tissue type is fine. But I feel like you want to do something that's actually like a little bit more internal, maybe doesn't divide super often, to kind of decrease the chance of replication related mutations, not just occurring, but showing up, right? Getting those mutations. Play. Play? Wait, what did he say? Oh, yeah. Dolly was mammary epithelial cell, absolutely. I think she also had health problems later in life, and I don't know if they ever quite knew if it was because of the way she was created. I'm sure somatic cell nuclear transfer technology is better than it was back in the 90s, but I actually don't really know what the literature on that, like what the things that you need to consider for to get a good clone are. I'll check that next, or I'll check that now before I forget. PubMed. Somatic cell nuclear transfer. Failures, successes, and the challenges ahead. Give me that free text, please. International Journal of Developmental Biology. Huh. This seems like oh no, immediate accepted article publication. Huh, okay. I don't know if and this was written in 2019. I just don't know if I trust this journal. We'll open it and see how we feel. Oh, my cat's in my room. Psst, come here. Maybe she's not supposed to be in here while I'm streaming, but maybe she'll come on screen. If I ignore her and pretend like it would be inconvenient that she would come over here, then she would come over. You've cited an article with the exact title? Really? Wow. Okay. Let's see. Okay, so they got some details. Oh, no. Look at the formatting. The male gamete is the perfect nuclear transfer device. Oh. Oh, no, no. Oh, no. In this paragraph, we will address the limited... Yeesh. This is not written super well. I don't think I want to... Yeah, no. The only thing I'll trust is that current efficiency hampers its practical application. Strategies put forth to improve its efficacy have been negligible in effect in farm animals. Okay, I'm gonna just believe that and call that good. That's probably not a very good article. '25th anniversary. Oh, well, okay. I'll check this one. Check this one. It's in something called reproduction. Hopefully that's a little bit better. If it's behind a paywall, I'll actually feel a little bit better about it. Let's see. Oh, that's cool. The number of publications since the production of Dolly, and specifically on somatic cell nuclear transfer. So we're doing mice, rabbits, sheep, cows. So these small mammals, those might be laboratory mammals. Livestock. So pig is probably also sort of a laboratory mammal. Tiger. Okay, that's kind of cool. Okay, I know I'm getting off track, but I can't help it. Somatic cell nuclear transfer. Tiger. Oh. It's a real journal. Reproduction is a real journal, right? I just assume it's not a great... Oh, you're wondering if that article is an older article republished. Oh, that's interesting. Yeah. Look that up. Sorry, here, I will copy this and put it in Discord so that, or actually I'll do the full text link. I'll put this in Discord so you can compare if you'd like. There you go. Okay, it's in Discord, but we don't want to look at that one. I want to learn about tigers. What do they do? Interspecies nuclear transfer. Theriogenology was another journal used a lot for assisted reproduction technology. Interesting. Let's see. They compared the ability of somatic cell nuclear transfer, or somatic cell nuclei of the Siberian tiger in pig cytoplasts and to support early development after reconstruction. Whoa. That's cool. They're explaining their experiments in the abstract, which is kind of weird. Skip to the results. After nuclear transfer, they did not see any significant differences in the development of tiger porcine reconstructed embryos. Strange. But they didn't keep them going. So I don't know if it's clear, but the reason why you remove the nucleus is because the nucleus, or the haploid nucleus that's inside of the egg, and the sperm, but we don't care about the sperm. The sperm doesn't really do much. The egg is where it's at. That nucleus doesn't actually do anything for a little while. So after fertilization, it actually takes, depending on the organism, several hours to actually begin to use the genetic information that forms the embryo. The initial work that's done by the cell to begin the process of embryogenesis is completely done by proteins and RNAs that are then probably transcribed into or translated into proteins that were put in the egg by the mother. So for the first set of cell stages, the first cell cycles of the emerging embryo, completely controlled by the mother's genome, which is, really fun to think about. So I'm actually surprised. I mean, I'm not gonna go much further into this, but I'm surprised that the pig cytoplasts supported early development with Siberian tiger nuclei. I don't know. I guess maybe all the maternal products from the pig could just keep things going well enough until it needed to turn on that embryonic genome, and then it all crashed and burned. But I guess there was enough conservation so that the pig could, I don't know. That's strange. I'm surprised. Your 20 papers about sperm you're digging through beg to differ. Yeah, I meant, you. Maybe you don't know what I meant. I know what I meant. No shade. It's just that, when we're talking about the first couple stages of embryogenesis, obviously you need the sperm to kickstart the whole thing. You certainly need that paternal genome if you want a diploid embryo. But those first couple stages, it's all about the mother. It's all about the egg. All right. What was I doing? Right, this. Maternal specific. Okay, so they're just talking about just kind of skimming. Let's see, the current strategy and state of art of SCNT in large animals. You do all this, you look at the genome, and only about 0.4 to 1.7% actually develop to term. That's interesting. That's really interesting. So parental specific reprogramming in gametes. I'm not sure exactly what they mean by that. Maybe what this, let's see. Huh. Oh. I don't know what this means. I feel like they're kind of suggesting that they might have to reprogram the maternal and paternal components from a previously diploid cell in order to have more success at developing them to term. That's interesting. So, like, you basically take a cell, like an ear cell. Wait, no, an ear skin cell, that's what I meant to say. Separate out the maternal and paternal components that were separated until fertilization, reprogram them through maternal and paternal specific pathways, which is what happens when you create a sperm or egg, and then recombine them? Huh. That could be interesting. Well, that's just what I'm taking away from that image. All right. Scans, welcome. Why do 90+% die mid-gestation? Honestly, I don't know for certain, but I would probably guess by what I was just skimming is sort of what I was also related to. The process of fertilization kickstarts a process of control by the maternal genome, and then a handoff to the zygotic or the embryonic genome. Now the thing is, sperm and eggs, even though when they combine, they become a single cell that is what is called totipotent or omnipotent, which means it can become any cell, like any cell. Even though that's true, the sperm and the eggs themselves are actually very, very what we call differentiated. They have such specific jobs. The sperm genome, for example, is compacted into these things called protamines that are not really found in any other sort of DNA packaging. So they're both sort of set up in a very specific way so that when they come together, they do something. When we do somatic cell nuclear transfer, we're taking a genome that isn't packaged like that, and we're shoving it into a new environment where we hope it, and it does, activate, the process of development. But probably because it's not sort of made for that purpose, a lot of things probably can go wrong. And honestly, you probably just get lucky with that sort of, because it's not just 90% die, it's like 99%, 98% die mid-gestation. And probably that success rate is like on the very early stage. I'm sure once you get past a certain stage, it's more likely to survive. There's also the question, which I don't really want to go into, but I do want to know generally, which is like, if you're taking a skin cell, like of my skin, for example, if you're looking around for a cell that's likely to have some mutations, skin cell is probably one that's accumulated more mutations than other cells. And I say that only because of its proximity to the giant ball of radiation in the sky, right? Now, that's probably not quite true, but depending on the skin cell, the cell type that you take it from, you're kind of shoving a square peg into a round hole, and that square peg already has some scratches on it, if you know what I mean. It's a little dinged up. Oh, okay, great. Yeah, you're welcome. I love explaining things. I hope that was correct, too. I'm pretty sure that's correct. And Mr. Frizzle, you found this old study about transgenic cows edited to have better immune responses to tuberculosis. Oh, send it along. I'd love to see that. Anyways, we are 2 minutes through this video that I started, 45 minutes ago? Maybe only a half hour ago. So that's all true so far, to an extent. Genetically similar, yes. Identical, yes. Probably barring the odd mutation, but you actually can get individuals that look differently. So cloned cat. Let me look this up. Aw. Let's see. I think that's this one because I recognize this picture. So, for example, these are two cats. This cat is the original cat. That cat is the cloned cat. And if you notice, there's a little bit of a difference in their coloration pattern. And that's because not only do you have to worry about the actual sequence of the DNA, right? You also have to worry about the epigenetics, the epigenome, the chromatin on that DNA. And just because of how cat coloration is is sort of determined. The cloned cat is not calico, even though its DNA was taken from a calico cat. Also, yes, calico cats are so cute. Yep, and you've got it correctly, or you've got it correct. X inactivation is the sort of answer to this. So it's very likely that what happened was that the cloned cat, it's one of its chromosomes, which encodes for coloration, was already inactivated, and it sort of shows you that even though you stick this nucleus into a egg cell that goes through its whole own stage of development or stages of development, it still remembers what it used to be. I forgot about that too. But yeah, he's right. Unless you are cloning a genetically female organism and used its own egg to clone itself, which would be kind of cool. Yeah, you have different mitochondria. That's the worst mitochondria. Oh, okay, he stopped on the mitochondria. He's absolutely right about mitochondria, but I do wonder. I don't think mitochondria differ that much between people. I just don't know the differences because mitochondria are pretty conserved. There are differences between people. You can measure lineages between mitochondrial DNA, so that's you know, matriarchal lineages through the mitochondria, just like you can watch lineages of men, or patriarchal lineages through the Y chromosome. I'm just curious, what is the mitochondria? What am I trying to say? Mitochondria mutation rates in humans. Let's see. So, wait, what? I couldn't tell what that was. Oh, here we are. 10 to the negative 8. Is this mitochondria? Yeah, 10 to the negative 8. But then we also need to know human mutation rate. Okay, actually 10 to the negative 8. It's on the same order of magnitude. All right. Never mind. I guess they can sort of change. Or maybe the number of alleles in mitochondrial genes. Yeah. Oh, I see what you mean. Mitochondrial. Yeah. How would you say that? Allele number? That's probably not not great. I mean, yeah, there are only 37 genes. Just on average, there's less stuff to mutate. I guess if we look at SNPs, maybe? Visualization of human mitochondrial SNPs. I would sort of assume... Oh, this isn't super... This isn't super helpful. Actually, do I have any in my DNA? So this, let's see. Column 2 is chromosome. I don't know if chromosome M is here. No. Yeah, I feel like this is... Yeah, I don't know if I want to go down this rabbit hole. Huh. Anyways, there are differences between people for mitochondria. So that's still, yeah, that's still absolutely true. Oh, play. Or not. Please. Please. Yes, and before that, I forgot. I did pull up that link about transgenic cattle. So let's see. What did you say before? Transgenic cattle edited to have better immune responses in tuberculosis. Yeah. So this is probably similar to what Dr. Z was telling us about, where they were doing genetic editing of pigs, that they made via SCNT to make their organs more human-like for xenotransplants. So let's see, what do they do? They wanted to assess the effects of, we'll call it IPR1, intracellular pathogen resistance 1 transgene. So where's that from? On preventing infection. So transgene just means that they are putting the gene in, but not in the place that it might have been if this gene is found in cows already, even if it's a slightly different variation. And prevent Mycobacterium bovis, which causes tuberculosis. They made a specific expression vector for the IPR1 gene, and they inserted it between surfactant protein A and methionine adenosyl transferase 1 of fetal fibroblasts. Then they did cloning. They cloned it. And then they found the blastocyst developmental rates. Oh, cool. And they actually had some surviving ones. They had 4 surviving and 1 dead. So actually their rate was pretty good. Well, actually, I don't know how many they actually made, but I assume they didn't do hundreds and hundreds and hundreds. So maybe the success rate for cows is just pretty good because we know what we're doing. They did some stuff, and they found that the transgene was in the right spot. It didn't affect the expression of surrounding genes. Okay, I'd like to see how they actually figured that out. Oh, cool. And they basically made them slightly more resistant against tuberculosis. That's actually pretty cool. Let's see, you don't have access. I probably don't have access either. Oh, I have access. No, I don't. Can I access through another institution? Hold on, gotta log on somewhere else. That one. Let me log in. They sent me a folder. They sent me a link. Where's my phone? Ah, here it is. You have a paper copy? Did you literally pull out your old folder or whatever of your, of everything you use in your bibliography or your just references? What? Yes, I... Oh, Brian, you saved me. I almost had it, though, I will say. I almost had it, but thank you. Cool. Oh, are you still looking for? You're still looking for that other one. Yeah, yeah, yeah. All right, let's look at their. Oh boy. Ooh. When was this? 2015. 2015 is. Um. You stepped away, so you were slow this time. That wasn't slow at all. I mean, I was not even to getting the art. I could have gotten that one because I have access. I just had to open my email. Um. So this was clearly done prior to CRISPR. Uh, I mean, sorry, I made that sound because like 2015 isn't that long ago, but it's just long enough ago so that they're doing some older techniques. Um, yep. So they're doing some old school cloning. Oh, they have some nice little cells, I guess. Showed some chromosomes. I've always wanted to get a karyotype. Yeah, the figures are older looking than 2015. They're giving more like 1999 like vibes. How about I zoom in? Yeesh. So this right here is called a karyotype. Something I've always wanted done on myself. So these are the condensed chromosomes of a cell, because cells don't normally have their chromosomes condensed down. And then they take a little picture of all of them, and then they align the pictures all together so that you can compare the homologous chromosomes of each of the chromosomes. Down here is the X. So I guess this is a female individual, and then we have 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6. Yeah. So I guess cows have 30 chromosomes. Did I do a PhD? Yes, I did. I have a command for that. I have doctor and PhD in my name. But yeah, no, I did a PhD. My PhD is in biochemistry, but really I'm a molecular biologist. Hold on, I have a command for this. CV. There you go. That's me. It's a command I forget about. Another command I forget about. I don't know if this will work. It didn't work. Whatever. What was I doing? Yeah, so anyways. Okay, that's fine. I don't want to read that. Oh. Oh. That one works. Does protein work? No. Why didn't genome work? Whatever. Oh, you're a biochemistry student. Awesome. Yeah. Like undergrad, grad school. Biochemistry is pretty great. Biochemistry has a pretty wide range of topics. I would not say I'm an old school biochemist. Oh no. Okay, so their primers are in table 1. Oh, cows. Oh no, is this a dead cow? Oh. Well, at least we get like 4 happy cows. And then 1 cow that didn't come to term. A, what is A? Oh, okay, so they actually got quite a few blastocysts from this. So that's pretty cool. So let's actually look at their data. So they were looking at the relative expression of these genes. So so this is methionine adenosyl transferase, and then SP. So it was like surfactant something something or other, surfactant protein something. I assume. Actually, I shouldn't assume. We could just go look. No, they don't. Oh, they do. Yeah, yeah. So they. I see. I didn't understand this organization before. So up here at the top, they are diagramming out that in the genome there's this SPA, MAT1A, two genes. This is their construct that they made, and they popped it right in the center. So they just plopped this gene down right between these two. Interestingly, they've replaced it. Oh, long arm, short arm. Long arm, short arm. Okay, those could have been homology arms, actually. Yes, yes. Okay, so they didn't replace it. That's good. So anyways, they want to test to make sure that by plopping that down in the center, they don't mess up those two proteins on either side. At least on the expression or the expression level on these transgenic and the targeted ones, they're not really very different between the two. And then they tested a couple things, which was apoptosis. So they were testing apoptosis versus necrosis. So I think necrosis is the result of a tuberculosis infection or other infection. They might have done other infections. And when you're getting necrosis, that is not a good thing. That means that not only are your cells dying, but they're dying in a way that is very damaging for the rest of your body. Apoptosis is a controlled cell death and can be indicative of a more controlled response to your body against disease. So what they're showing here in A versus BBB are that transgenics who have greater amounts of apoptosis after infection relative to the control, which just is necrosing. And then, oh, cool. And then when they do some cell culture or something, they're checking how many of the Mycobacterium bovis cells survive. And when you have the transgenic cows, they mostly die. Or not mostly, they die more. They don't have metrics, but this probably is significant, just based on the error bars. Well, cool. That's a cool little study. Thank you, Alt Miss Frizzle. Yeah, that was fun. Yeah, you guessed that apoptosis would be a result from an immune response, say from cytotoxic T cells. Yeah, exactly. I'm not very good at immunology, but that's more or less probably what's going on. Usually, just I think it's pretty safe to say that in response to injury, disease, some sort of invader in a body, you never want necrosis because that's just rotting tissue inside of you. You want your body, if it needs to deal with something, to do some apoptosis to sort of clear it out. In fact, I even know like some cancer treatments nowadays are so effective that you have to be very careful with them because you can get, I think it's called necrotic tumor syndrome. It's where you kill the tumor so fast that it rots inside of your body, which is bad because you can potentially die from that itself, even though you've effectively killed off the cancer. Yeah, thank you, Miss Frizzle. This was cool. A very succinct, good little show of what we actually can do with cloning and also genetic editing. You could basically combine the two. Back to Metal Gear Solid. Oh, my cat. She's joined us. You still don't understand how that protein confers some sort of resistance to tuberculosis in cows? Me neither. I kind of just skipped over that part. What was it? IFR1 gene. Let's look it up. But yeah, the transgenics was really cool. Wait, was it interferon? No, that wasn't right. I'll open it back up. What was it? IPR1. Gene. Oh, well, here's a study. An earlier study implicating it. So let's see. So it limits multiplication of a tuberculosis strain and switches the cell death pathway from necrosis to apoptosis. That is cool. But yeah, what is IPR? IPR1. Like, what's its role? See, we might just not know the function of it, unfortunately. Although this is from 2005, so that was over 15 years ago. Its name is intracellular pathogen resistance. Okay, I'll look this up again. Huh. There's not a ton. I just skimming these, it kind of sounds like not a lot has actually been done. Like, we know the overall effect, but not specifically how it works. Let's go to Uniprot. I also don't know. Well, because how big was this? How big was the gene? The gene itself. IPR3. Oh, okay, I don't know if this is probably not to scale. Wait, it's very strange. It's not coming up with oh, it has a different name, I see. In mice. May act as a transcription factor. Plays a role in innate immunity against intracellular pathogens. Maybe Protein Data Bank has a structure of it. That's a good, yeah. Promotes p53 function on certain genes. I mean, if it's a transcription factor, then it might just control just a general upregulation of any number of immunity, pathogen response, that sort of set of genes. So it might actually not do something like functional. It might not ever actually interact. IPR1. It almost seems like they figured it out through studying vaccine response. I didn't see anything vaccine response just there. Oh, hold on. I'll be right back. My cat wants out of the room finally. I guess behave. I'm back. And sorry, Brian, that was here, let me do it for you. That was not activated on that screen.
Boo!
There you go. Yeah, so it looks like it's a transcription factor. The structure was a good idea, but I don't think it's actually there. Yeah. Okay. So it's a transcription factor. It's just so, for those who don't know, transcription factors are proteins that go around, they sit on other proteins, or, excuse me, on the DNA, and they either turn on, turn off, modulate expression of genes. So genes don't just know when to turn on and off. So that they need other little helpers that maybe perhaps take input from even more proteins. So you could imagine that maybe IPR1 is one of these like intermediates, maybe some other extracellular, some signal from outside the cell comes in, turns on IPR1. It's like, okay, stand back. And it goes through the genome and turns on all the genes that will help fight an infection. That might be what it is. Okay. Let's see, what were we doing? We were just talking about more about IPR1, right? But really, we're still trying to get through this this video about cloning in Metal Gear Solid. Well, I don't know. That's something I feel like is pretty important to talk about. Oh, hey, Defested. Welcome. I hope was it bathroom renovation? You were doing something home improvement wise. No, you were doing something in your basement. Hope that's going well. I saw you were on the other day. I just couldn't join, unfortunately. Adding a basement bathroom. Nice. Oh, actually, adding a full one. I thought you were renovating one that already existed. Well, I'm glad it's going well. That's awesome. So this video came out in 2014. I'm trying to remember. So this is sort of wrapped up in the like stuff that's has been going on in China, where they've been doing some work in genetic editing of human embryos. What I don't remember is if any of those resulted from cloning. See, I do remember human cloning China. I just don't remember. Yeah, there were CRISPR babies. Okay, maybe it wasn't cloning. Okay. Yeah, I mean, like, the ethical concerns are, I feel like, I don't know, this is sort of interesting. There's an argument to be made that human cloning, if perfected to a degree, could be used for reproduction for people who cannot conceive or have children otherwise. So, for example, single people who want to have a baby that is genetically related to them, well, they could, in theory, rather than seeking a sperm or egg donor, just get themselves cloned. Human CRISPR was on CCR5, and that was on embryos that were going to get used for in vitro fertilization. Thank you for the clarification. Yeah, we talked about the CRISPR part last time. I just wasn't sure. I was getting confused whether or not they had done SCNT. Anyways, I've always liked the idea of, or been fine with the idea of people using cloning as a means for reproduction, just because it's not going to be like so widespread that suddenly we're all just clones walking around. That probably will never happen. People like to reproduce way too much for that ever to be a huge problem. And like you can say like, oh, it's like the height of, hubris to just clone yourself. Like you can imagine like rich people just squirreling away their genetic material and cloning themselves over and over and over again. But each time it's a different person. I missed. D&D ended early for you, so I got to catch part of the stream. Awesome. Sorry I missed that. And the transgenic cow paper was from China. I did notice that. I mean, China does a lot of great research. But there's like one or two or three, big name biotechnology people, molecular biology people in China who have done some things that not everyone else has been a fan of. So cloning. Also, I guess you could. We wouldn't. We wouldn't just get rid of one of the 4 Fs. Yeah, exactly. Feeding, fighting, fleeing, and mating. Yeah, no, exactly. Like, I feel like the argument against cloning being that people will do it too much, that's never going to happen. People are not going to stop having sex with each other. That's not going to happen. I mean, we're like 1.8 billion years into life on Earth. Like, whenever sexual reproduction evolved, like, we haven't stopped. So, or at least not, our lineage has not stopped. So that's not going to change. But yeah, there's other like means. So like, there's also like cloning adjacent things. So I'm trying to think. No, this wouldn't be cloning. I was just thinking about whether or not the idea of having a same-sex couple, either male-male or male-female, and even by that I mean sperm donor, egg donor, whether or not you could have genetic children between the two. I feel like egg and egg, that's a little easier. Sperm and sperm, you still need an egg donor, but then if you remove the nucleus from the egg, it's not SCNT, because the S stands for somatic. Those are technically germ cells, but I guess it would be GCNT, germ cell nuclear transfer, if you want to have a 3-parent child who is, genetically the product of 2 sperm donors, and then they have the mitochondria from the egg donor. I wonder if that exists. Germ cell nuclear transfer. Birth of mice produced by germ cell nuclear transfer. Okay, well that probably doesn't mean exactly what I meant it to mean. So that's in 2005. They were just doing some. Oh, okay. They were basically trying to figure out cloning. Oh, Mr. are you Googling at the same time I am? Let's see. What would I say? Like sperm cell nuclear transfer. Two dads. Hey, how researchers made mice with 2 dads. Nice. They're so cute. All right. 10 amazing things scientists just did with CRISPR. All right, show me the data, please. Generation. Oh. That's a new word, bimaternal and bipaternal. Generation of bimaternal and bipaternal mice from hypomethylated haploid ESCs with imprinting region deletions. Oh, so you're just thinking this may cause a problem because of your issue with your molar pregnancy. It doesn't make a baby when the father's chromosomes are duplicated, but maybe that is just from the same dad and not two different dads. Let me think about that. Also, I do not remember. Let's see. Okay. Didn't remember exactly what a molar pregnancy was, so I had to look that up quick. So it doesn't make a baby when the father's chromosomes are duplicated, but maybe that is just from the same dad and not two different dads. Yeah, so this is definitely a biotechnology technique. So you also have to have that little like grain of salt like attached to this, where there are interventions that maybe a improper sort of pairing or coupling between gametes doesn't produce. So in this case, or maybe not in this case of what I'm showing, but in what I'm talking about is, yeah, two different haploid sperm cells from two different fathers. Those would probably have to be removed in some way from the sperm and physically relocated into the enucleated, or the cell, the egg cell lacking a nucleus, to sort of kickstart everything. There are specific molecular barriers to multiple sperm cells fertilizing the same egg. But if that is overcome and something weird happens, that can result in termination of pregnancies. I think it actually happens more often than you think. And that's because of differences in chromosome number, where you're not haploid or diploid. You are something in between or something more than diploid. Those cells are non-viable, and those pregnancies, they don't just come to term. They don't really begin. So they're also researching whether you can reprogram primordial germ cells into sperm or egg, so you can have the opposite gamete needed. Oh, that almost seems easier, right? That seems way easier. Although I feel like, okay, making sperm cells, that's got to be easy. Making an egg cell, though, I bet that's hard. You need all those nurse cells, all that stuff. I'm willing to bet making sperm cells are easier. But yeah, that's really cool. Wait, so let's think about this. So, if you are a person, a human person, and you got the opposite gamete made of whatever your body naturally makes, both of those gametes would undergo meiosis, and part of meiosis is homologous recombination, or crossovers. The crossovers happen. Meaning that you would get a random assortment of the chromosomes, and even though they're from the same source, if you ended up putting them back together, you wouldn't get a clone of yourself. You potentially would get a more sickly version, just a sickly child, because you're increasing the chances of basically inbreeding, self-inbreeding. Am I thinking about this correctly, or am I rambling? Fibroblast to embryonic stem cell to primordial germ cell is doable for sure, and it's been done. Oh, that's cool. And I know what you're saying is like probably for like, male-male or female-female couplings to have sort of a different style of fertilization between the two gametes. I know what you're saying is that, not what I'm saying, which is, well, what if you took the gametes from one person, split them off into two, and then recombine them together? I think that would be a bad thing. Yeah, because you will never increase the chance. No, you could. No. You never can increase the chance of heterozygosity. You will only increase the chance of homozygosity. Or stay neutral. Yes, thank you, Defested. You're absolutely correct. Most teenagers try to breathe with themselves sometimes multiple times per day. Yep, that sounds about right. Absolutely. Oh, I will hydrate. I don't know if people can hear that one. I hear the words hydrate being said to me, but I feel like that one doesn't play for people. Oh, anyways, okay. Getting so off topic, but that's okay. So what do they do here? Ooh, androgenetic. So this is a word I knew. Parthenogenetic, or parthenogenic, meaning like from only the female. I've never heard of androgenetic. Yeah, so so PGC, like methylation profiles, there is a problem when you're doing like cloning of different types, or so the genomes of individuals in gametes, or the genomes of gametes, are imprinted. So they actually differ between the male and the female, and these are done through epigenetic means. And these imprinting regions are kind of important. I just don't know too much about them. So it sounds like they deleted them. they just got rid of them so they didn't have to deal with it. And it produced full-term bipaternal and bimaternal mice. Ooh, an image. I might have clicked on it. Ooh, I've got something open that might be making my computer angry. All right. So they took gametes, they made haploid embryonic stem cells. So that's interesting. So they kind of push them towards embryogenesis. I don't know how they did that. And then they allowed for global demethylation, making imprint-free ESCs. So that's important, because if they are imprinted the same way and brought together, the pregnancy will ultimately fail. I think it's not designed. It's not the purpose. The evolutionary purpose is not to keep two sperm cells from making an embryo, because that can never happen. But a result of them is that it wouldn't really work out very well. So basically they took these, they must have combined them in some way. Haploid. Huh, okay. Very cool. You recently did a presentation about DNA methyltransferase, which required to establish the maternal imprint in oocytes. Yeah, absolutely. Yeah. DNA methylation is basically the major pathway. I think there are some histone modifications that are important for imprinting. I just don't remember. And I feel like it's more of this thing where it's like, well, we're pretty sure that it's there, but we're not really sure if it's like how big of a deal it is. It is a lot about that DNA methylation. So that is methylation of the DNA molecule itself, not the histones. Although if someone told me that there was imprinting on histones, or due to histones, I wouldn't be surprised. I just don't remember right now. All right, what are they doing here? What'd they do? Give me another cartoon, please. Oh. Histone 3. Oh yeah, H3K4. That makes sense. H3K4 is a big one. I guess let me just look that up. H3K4 imprinting. Wow, you joined stream on just the right day. Don't usually talk about imprinting. Let's see. Oh, this one, oh, interesting. This is just saying that huh. Okay, that's not related. Histones are super important with regard to genomic imprinting. Not surprised. I should know that, but that's not exactly what I study in terms of epigenetics, so I don't think about that very often. That's good to know, though. I will definitely have to read more about it. Oh, get a link to a paper. Anyways, I wanted to make this. Oh. Oh. Is that what they look like? They look like little hippos. Oh. Oh no. They exhibited evidence of increased inner pressure, including unclosed eyelids, leaky guts, and longer tongues. Yeah, they look like little potato mice. I don't think they're... Okay, so I think they went to full term, but I don't think they're very happy. Poor babies. Oh. See, this is why I don't work with mice. I just think they're too cute. Even their little pups. Oh. I hope at the end they just say that they all lived. Okay. Oops. Okay, so they were doing some... I did see, though, that they were trying to ameliorate some of these defects. So that's kind of cool. So they're changing some of... Oh. Look at that. This pup looks a lot better. See, that's what I remember mouse pups looking like. Okay. So they were ameliorating the bipaternal mouse defects with a GNAS region defect. So what is GNAS? Oh, so it's an imprinted region specifically that they found in these embryonic stem cells. Okay. Wow, it's just this little tiny region that they deleted. No, it's not this little tiny region. It's a much larger region. Okay. Yeah, just looking. Ooh, yeah, so I guess I feel like just some sort of trigger warning. Don't look away if you don't like to see bisected mice. But oh, yeah. So this is a cross section of a mouse pup that have all been born, I'm assuming. Wild type, and then so they had their first version, the 6KO. Oh yeah, look at that. Yeah, so what these arrows are pointing to are different defects in these mice. I don't know mouse anatomy too much. It's nice that this wild type is here. So they're pointing at the tongue, which is huge between the two. Something wrong with the gut, and then there's this distension up at the front. So you can see, oh, actually, can you see my mouse? You can barely see my mouse, but this right here is the front, the sternum. Look at where it is on this wild type mouse. And there's, right, it's a baby mouse pup. They don't really have, they're very slender because they don't really have a lot of body fat because they were just born. It's basically just bone and then skin on top of it. But this one, there's this huge area that's just distended. So that's why they look like little potatoes. Also, they don't point it out, but its snout is more or less missing. I mean, it's very deformed. Poor thing. Meanwhile, if we look at so this is their 6KO, so this is version 1. Their 7KO has this extra deletion. It looks way more similar to wild type. So that's really cool that they were able to like ameliorate these problems related to genetic imprinting by deleting this one region. Well, I don't know if the 6 to 7KO refers to multiple deletions that they had. Okay. Oh, yeah, it is. Yeah, sorry. I went ahead too far. So they started with androgenetic haploid ESCs. So these are embryonic stem cells that are made using only male like germ cells. So this is like a sperm that was coaxed into being, at least I think it's a sperm cell, some sort of male haploid cell, coaxed into being an embryonic stem cell. They deleted 6 imprinting regions, so that's why it's KO6 or 6KO, so 6 knockout. They found those. They co-injected it with a sperm, so that's really cool. So they took their imprinted ESCs, which were haploid. They deleted the imprinted regions and injected that into an oocyte or a premature egg, essentially making their own egg that was no longer imprinted as male or a sperm. And then they also injected a sperm. So now they have androgenetic diploid ESCs, and then they had to do some like clever cloning, more or less. So this one is where they take this stage of development is where these cells will become the extra embryonic tissue, so the tissue that becomes the not the uterus, placenta, whereas the cell that they're injecting will all be their androgenetic diploid. Imprinting genes are usually massively important to embryonic development. Absolutely. I mean, the imprinting helps define some of the initial stages of embryonic development and later stages of embryonic development between the two chromosomes, because the chromosomes will they won't even out in terms of their epigenetic landscapes, but their epigenetic landscapes will change during the course of development, which is important. But you're absolutely right. The contribution of each, in a way, is measured by the imprinting done of them. Were you going to link to a paper, by the way, Scans? You said this is an amazing paper by Saccone. Oh wait, is it this that I'm looking at right now? I didn't look at the authors. No. I just didn't recognize that name because I don't study this. All right, where were we? Yeah, so unfortunately in this 6 knockout, yeah, they got these sad pups. Oh, they're so sad looking. But yeah, then one more deletion, they fixed it. That's super cool. Anyways. Oh, you were looking at the one I was looking at. I'm sorry. Not paying attention. Yeah, let's open this up. Actually, now that I'm reading this title again, this sounds very familiar. I feel like I have read this at some point. It was cited a bunch. Yeah. I must have read this at some point. This one I think was weird the way I was reading it. It's like, this protein that I think was related to a histone demethylase, unsurprisingly, has a non-histone target, which happens to be potentially DNA. I don't know if they show here directly DNA methylation. The results suggest that demethylation of H3K4 is critical for establishing the DNA methylation imprints during oogenesis. Cool. That's interesting. Yeah, because all these different marks sort of have, well, not all of them, they can have sort of cross-talking mechanisms. Oh, Megan, you think you've read this? That's cool. I will link it in the Discord. We've built up a lot of Discord links today. Okay. Gotta finish this video. That's what I was saying like an hour and a half ago. I mean, it was obvious that's where this was going to go because that's the easy answer. And that is assuming, as Scans has brought up in this conversation, assuming that the imprinting has no effect or has doesn't have like too big of an effect, such that it alters the way that development starts from the very beginning. Because the environmental factors he's talking about, I guess technically, also includes the epigenetic environment that the gametes that made you were exposed to. You're going to clone your cat? I mean, that's a service. That's a service you can do, or you can buy. Or at least you used to. Clone my cat. I think it's very expensive. Ooh, Viagen Pets. Pets and Equine. Barbara Streisand cloned her dogs. Oh, I mean, why not? But at the same time, I would be so sad. I would miss my cat, plural, my cats, and I would just be sad. It would just remind me that the cats I loved died, and I replaced them with Frankenstein monsters. Not really, but I replaced them. You know what I mean? I feel like I would just want to get a new cat with its own personality. Start genetic preservation. Yikes. Give me how much it is, please. Initiate cloning. The total cost of cat cloning is $35,000, also paid in 2 equal installments. Oh. Subject to state tax. Initial payment. Oh, why do you have to put in this? That's so sad. Oh, I feel like this looks like it's a scam. I'm sure it's fine. They have merch. I cloned my cat at Viagen. No, they don't have that. Whatever. Okay. Oh, telomeres. Yeah, right. Telomeres. Telomeres. They're both like really important, but also not the smoking gun. I'm pretty sure we might have looked at this, but I thought I had read that there's just conflicting information about whether or not lengthening telomeres actually does anything for long-term viability and health. Like, it seems like, yeah, it's super important. Like, keep your telomeres long if you want a long-living cell. But at the same time, just lengthening them isn't really sufficient for an immortal cell. All right, we're done with that. Cool. It only took us an hour and a half to get through the entire video. Cool. Oh, you sent me. Oh, a woman cloned her pet after it died, but it's not a copycat. Oh. Okay, wait. So this person's cat died in 2017, but there's nothing that the person could do. Oh, and she talked to her roommate about Viagen pets right after the pet died. So she called them. $25,000. Wait. Was that cheaper? Yeah. She has the carbon copy of Chai curled up in her lap. Belle is nearly identical to Chai. Really, but the similarities end at like the physical and also stretching out when they sleep. Huh, that's interesting. There's a shortening of telomeres, but also the accumulation of reactive oxygen species. Yeah, absolutely. Reactive oxygen species cause all sorts of havoc with things that you don't want them to be wreaking havoc on. So they mess up a lot of proteins, and proteins being messed up, even though you can sort of clear them out of your system, that does mean a increasing nosedive of cellular health. But if you start also messing up your DNA, that's where you're going to get into some trouble. Yeah, there's a bunch of things related to aging. You recently lost your kitty. Oh, I'm sorry. And it would not make it better for you. It's going to be a long time before you get another cat. Yeah. I have 2 cats now. They're fairly young, thankfully. But I recently almost a full year ago, I lost my rabbit, who I had for like 6 or 7 years. And it's going to take a while before I get another rabbit, probably. I don't know. We'll see how I feel in a couple months. But yeah, absolutely. And yeah, 25,000 is a lot. So then they go on to explain it, and then they have a surrogate. Then you get your clone back. And then they mention that it's not always a success rate, just because it's a pregnancy. So pregnancies are extremely fraught, even in the best of cases. Oh, cute. I'm just trying to find a place where oh, interesting. So Chai never got to experience the joys of kittenhood because she had illnesses when she was young. Oh, her first 5 months were spent under treatment. It's about giving her a second chance. Oh. Oh, and Barbara Streisand, yep. I'm just skimming this. I was just kind of curious about how different the 2 cats are. Yeah, I mean it's interesting. I mean, do what you want if you have the money, but I feel like it's uh you know there's lots of cats out there. There's lots of animals out there. I feel like maybe it's better to adopt one, but whatever. All right, so it is well over when I wanted to stop. Alt Miss Frizzle, yes, pregnancy, negative 12 out of 10 stars. Do not recommend. Yeah, I know you have personal experience with those troubles, so I'm sorry. But yeah, okay. I'm going to sign off for the night. Sorry for the extremely quick notification that I was actually going to stream tonight, but thank you for also joining. Yeah, join the Discord if you want to. Well, apparently not keep up because I don't do a good enough job of keeping people up to date. But now we have lots of a couple papers on there to look at. Oh yes, and then stick around for the raid so we can either go to Embis or Science Streams. We haven't got to Science Streams before. Let's go to Scott. Yeah, Science Streams. Yeah, let's head out there. So thank you everyone for coming. We will. I'll see you on Sunday, I think, where we'll be making more. Oh, thank you for the follow, Scans. Yeah, on Sunday, I think we'll be doing some more 3D printing of some little tiny itty bitty mazes for tardigrades and other microorganisms because that was fun. And I have my nail glue coming in. Thank you, all Miss Frizzle. Yeah, so thanks everyone. Stick around for the raid, and I'll talk to you later. Bye.